So my name is Steve Russell. I am a professor at the University of Iowa. I was involved in the initial phase 3 study, which was the pivotal trial to approve this Voretigene neparvovec or Luxturna. And I've been involved prior to the time that this was a company. We were involved as an academic exercise with the University of Pennsylvania, collaborating on this trial. So been with it for more than 10 years. Regarding this particular presentation, I've been involved primarily with Luxturna as far as gene therapy and other strategies for treating recessive diseases, primarily Leber's congenital amaurosis. So Luxturna is a complicated molecule, if you will. It is a true gene therapy. It's essentially a adeno-associated virus type 2, which has had its nuclear material removed and replaced with an artificial, what's called a transgene, artificial piece of DNA. And the gene contains the replacement gene, the wild type for the gene RPE65. And RPE65 is the gene that produces an enzyme called retinal transisomerase, which regenerates vitamin A to its active, photoactive form, high energy form, which is required for visual perception. So the way visual perception works is a photon comes into your eye and it hits one of these activated vitamin A molecules, which is sitting in a pocket of a protein called an opsin. It changes the conformation because of its energy yielding when the photon hits this high energy vitamin A. It then changes its shape. That opens a channel in the membrane and changes the electrical potential, which is then propagated through the system as what you and I see for each little bit of the seeing that we have with each photoreceptor. There's a system in place in the eye to regenerate these high energy vitamin A molecules in that they're shuttled out of the photoreceptor cells into an adjacent cell called the retinal pigment epithelium. They're then re-energized and then shipped back into the photoreceptor cells, primarily in the rod cells, which are the cells that detect light and dark. And they're primarily concentrated in the periphery, but there are some of them near the center of the vision. And in this particular group of patients that we treated, these are patients who have mutations on both of their genes for this particular enzyme, RPE65. And so neither copy works well, or probably minimally, if at all, in both copies. And so one of the requirements for enrollment in this trial was to have mutations in both copies of the RPE65 gene. And therefore, what we were trying to do is to replace that defective copy by providing additional copies to these cells, the retinal pigment epithelium cells, in the back of the eye. The reason I'm involved, and Dr. Al McGuire, who was also a co-author on this paper, is that in order for this gene therapy to become effective, the virus, which is like a naturally occurring gene injector, because it injects genes into cells, and in a natural situation, it would inject its own DNA into the cells, but in this case, it's injecting the DNA which we want to put into the cell, is a naturally occurring virus, which is known to be non-pathogenic in humans. And it has a large enough size that it can carry this particular gene and the activating portion of the gene called the promoter. And so that is placed adjacent to this retinal pigment epithelium. And in order to do that, we have to create an artificial retinal detachment. Vitreoretinal surgeons are needed in order to be able to deliver the drug to the site that's required in order for it to have access to the appropriate cell. And then this gene is taken up, transfected, essentially into the target cell, which in this case is the retinal pigment epithelium, and then it's activated and produces the enzyme that recycles the vitamin A. And these patients have a remarkable improvement in ability to see in dim illumination. They have a remarkable improvement in their peripheral vision, and they do not seem to have any deterioration of visual acuity, although we were unable to confirm in the original study that the visual acuity was improved. We did have some patients who improved, but there was a very high variability, so we were unable to demonstrate statistically that it was improved. What I was doing at this meeting was I was presenting the 8 and 9-year results. Now, the way the study was structured, the majority of the patients, 2/3 of them, were allowed to be treated initially, and 1/3 of them were held back a year to act as a control. And then at the end of that 1 year, when we had reached the determination of success of the primary endpoint, which was a mobility test, a walking test through an obstacle course called the multi-luminance mobility test, then the control group were then allowed to receive the medication in both eyes. And all of them did participate and receive the drug at that point. Based upon our measurements, they did equally well to the group that received the initial therapy, the original intervention group. And the problem here is that what we're trying to look at is the long-term sustained potential benefit of this agent. So as we went through the COVID era, we lost a number of patients in follow-up. So we wanted to include all of the originally treated group, and we wanted to add to those all of the patients who were in this delayed intervention group at each time point from the time of intervention. So we had to do essentially a time shift of a year so that they all lined up when they were originally treated. And we were looking at time points that were equal times from the initiation of the gene therapy. There had been previous studies looking at other types of gene therapy drugs, different formulations produced by others using not exactly the same DNA, different promoters, had shown that there was some effectiveness in one of the other trials, but it wore off after 3 years. So there's always been a concern about sort of what is the long-term durability of this gene therapy. Conceptually, it should last forever, as long as the cells don't divide and the DNA is not diluted, then it should essentially last forever. But, this is the first one that's been approved. It was the first gene therapy that was approved for an inherited disease of any kind, any part of the body. And so this is sort of the longest follow-up we have from a gene therapy treatment. And the evidence at the 8 to 9 year time point was that the durability of the effect on the primary endpoint appeared to be stable. Visually, they appear to be stable. There's a little bit more variability in the later time points because, again, we lost some subjects due to COVID and we couldn't bring them in and for full evaluation. The one parameter that did seem to be not as robust was the visual field. It did appear that there was more variability of the visual field in the later years, and perhaps this was not being maintained. There was another test that was done called the full field light sensitivity test, or the FST test. And that was sort of a measure of what's the dimmest light that someone can perceive. That moves in synchrony or with high correlation with the mobility test. And it also showed considerable durability with no evident fall off by the 8 to 9 year time point. So that's about the efficacy side of the equation. The safety side of the equation is pretty interesting too. It seemed that the adverse events were primarily concentrated in the early years, as you might expect, because this is a surgical intervention that's necessary in order to place the drug. We did have one patient who developed acute myelogenous leukemia and died at year 7. Malignant transformation is sort of a crucial safety signal when you're using gene therapy, because the question is there any involvement of the gene therapy agent or potentially insertion into the genetic material that leads to inactivation of a tumor suppressor gene? There was no evidence of that we could find, but nevertheless, that was reported as one adverse event. A second issue is the sort of the long-term safety concerns that have come up with this particular drug is that there seems to be some scarring that occurs in the retina in areas where we did not perform the actual injection. Outside of that area, there has been some scarring. Now, the underlying disease, which fits a syndrome called Leber's congenital amaurosis, can have that kind of scarring or similar scarring in the periphery. So it's hard sometimes to detect whether this is a new onset of a complication or whether it's simply part of the ongoing disease itself. But there have been some criteria systems that have been set up where this has been observed with higher frequency in the post-marketing study in post-marketing patients. And so we were able to identify 3 patients that met the criteria for this chorioretinal atrophy in the study. There were 2 additional patients that we found in the phase 1 study, which was not part of the reporting group that we were talking about here. So there have been 5 total of these chorioretinal atrophies that have since been identified in the original 2 cohorts that have been treated with Luxturna. Overall, the safety profile still looks pretty robust, and most of the complications appear related to the surgery and the procedure to put the gene therapy in the appropriate location so it can have its effectiveness, and not due to the gene therapy agent itself. Well, it has been safely rolled out in most countries. Not every country yet has access, but certainly most European countries, a few South American countries, Asia, just recently in Korea. So it has been recognized as a sort of a dramatic response, dramatically effective therapy for this particular inherited retinal disease. One of the issues, of course, that comes up is, can it be applied to any other disease? And the answer is no. This is truly personalized medicine. This is treating based upon an individual's genetics. In some ways, it helps to confirm the concept of a personalized approach to inherited retinal disease. It confirmed that we can use the adeno-associated viruses as conveyors for our gene therapy, and it was safe and effective in doing so. And it has generally excited the field of ocular genetics, and there are now a number of other trials. Unfortunately, none of them have reached FDA approval status yet. There are a few that are sort of knocking on the door. And so it's been sort of win-win-win as far as patient awareness. Patients are now much more receptive to being genetically tested so that they will be eligible for future trials and for future therapies. And clinicians have also gotten excited about the potential for being able to genetically characterize their patients so that they'll be eligible when therapies become available. Been a very encouraging drug group to work with. The Spark people have been very responsive to the weaknesses and the strengths of the drug and have been very forthcoming when there have been issues, basically trying to help as best I can in terms of getting these patients taken care of, because it really is a remarkable change that we see in these individuals who basically can change from being in a situation where they can't function in a day-to-day environment. And then once they've had the therapy, they're thrilled. Most patients with inherited retinal diseases would be thrilled just to stop their disease from progressing, let alone have an improvement. But these folks have, with 2 exceptions in the trial, there were 27 out of 29 patients improved, which is a number needed to treat of almost 1.0. So it's really been encouraging from that perspective.